Hi, my name is Alexa Alvarez, and today is July 23rd, 2020, and we are working on our microscopy for microbiology lab. And this video specifically is, I will be talking about the results of the lab. So with this lab, our main goal and task was to get to know our microscope, and part of what we had to do was identify the different parts of a microscope and what each part was intended to do. So when we received our microscope, it actually required a little bit of assembly, which, I mean, it wasn't much, but we had to put the objective lenses on there, and we had to pretty much assemble this, which this comes apart, and there's a battery in there. So other than that, it didn't require really much of anything other than just putting these, screwing these on here. We were supplied with an oil immersion lens with the immersion oil. And so what we initially had to do, like I said, was identify the different working parts of the microscope and really get to know it so that we know exactly, once we start looking at slides, we know exactly what part of the microscope we have to utilize to make sure that we're able to view specimens and cells accurately. So initially on the second, so that was the first part of the assignment. The second part required us to look at already prepared slides, which the slides that we looked at were, I'm just gonna read off the names because my slides are, I have to clean them. So we looked at amoeba, we looked at penicillin, we looked at yeast, we looked at bacteria spirillum, and we looked at bacteria bacillus and bacteria coccus. So just different shapes of bacteria, different strains of different bacteria. And from there, we were required to take pictures of the slides. So most of them, I think it was fairly straightforward. It's been, how long has it been? I want to say it's been maybe more than 7 years since I had last used a microscope. So one, I was a little rusty initially, so I needed to kind of work on getting to know this particular microscope. The ones that I'm used to at school are a little different, and so not that this is really any significantly different, but just being at home and doing this from home is a little awkward. But I was able to, on the amoeba, it was rather easy. Just the biggest thing that I found myself doing initially, I think on like the first 2 slides, was I was hunting around the slide quite a bit and looking. So even with the letter E that we had to look for and look at different magnifications, I was totally lost in there. Like I could not find it, and finally it came into focus out of nowhere. So that was probably my biggest challenge initially. Once I kind of remastered it, I guess you can say. I mean, I don't want to say master it, but once I got a little bit more comfortable, it was pretty simple. Taking a picture of the cells, that was a little challenging. So my phone it's one of the newer iPhones, so one of 3 cameras essentially worked, and it was kind of funny trying to figure out what angle and what positioning I needed to do and have a steady hand. So that was interesting. I know in the past, I remember having to draw, and I remember having to color and all kinds of stuff. So being able to take pictures and use technology is definitely a perk, I want to say. Kind of goes to show how far technology has allowed us to go. The only one I did have a hard time with, and it was a little frustrating, was looking and finding the coccus form. And that one, I could not find it. I mean, it was so challenging, and I had to, like, initially I was able to find a couple cells or a couple, I was able to find a couple of them, and I was able to zoom into the higher power. However, once I switched to the oil immersion, I would get completely lost. So I had to kind of work backwards and then drop the stage and then bring it back up once I switched into an oil immersion lens. So that one last night was probably my biggest headache, just because I think the cells were so small and they were hard to find. After doing the experiment today on the wet mounts on the cheek cell, there was kind of a little excerpt on there, like a little bold or side note to lower the lighting on the microscope. And I'm thinking back on how I was working it last night, and I think I might have benefited by lowering the light just to build a little bit of better contrast between the cells and the slide, or the stains, with the lighting. Other than that though, I think the picture that I got was half decent. Not very defined. I did find that I had a hard time really coming into focus. My fine-tune knob was tapped out. I was all the way to the most I can get, and I feel like I could have gotten a little bit more fine-tuning, but not entirely sure if it's just the microscope or if I needed to clean something in there. Not sure. With what we did today, so part 3 of our experiment was us creating wet mounts of both our inner cheek cells and our gum and tooth line to collect some dental plaque. So on that third portion of the assignment, I appreciated the picture that was included for the cheek cell. However, there wasn't one for the dental plaque. So I did Google, and just what I found on my own slide was very stringy, like filamentous cells. A lot of what's online is extremely magnified, and I'm thinking they're using completely different microscopes than what we're using here. I'm sure they're using very powerful microscopes to get that in depth into dental plaque. So I'm hoping that the image that I selected or that I took was a clear representation of what dental plaque is supposed to look like, just by looking across the lens and the slide, and seeing if there's anything different. That was probably the most consistent, and I didn't really see anything else that was worth zooming into in terms of cells or anything like that. Other than that, I think it was a pretty straightforward assignment. I rather enjoyed it. I think this is a part of microbiology or biological sciences that I truly enjoy, so it was nice getting back into it and working with slides and being able to look through a microscope and see different cells. So I think that's all I have for now. Thank you so much, and we will see you on our next assignment, probably later today. Bye-bye.